Hi, I'm Teacher Melai, and let's talk about the step-by-step analysis of biological data by John H. McDonald in 2014, so a faculty of University of Delaware. For his step number one, you have to specify the biological question you are asking. And then you have to put the question in a form of biological null hypothesis and alternative hypothesis. You also need to put the question in a form of a statistical null hypothesis and alternative hypothesis. Fourth step, determine which variables are relevant to the question. Fifth, determine what kind of variable each one is. And number six, design an experiment that controls or randomizes the confounding variables. So as mentioned here, you must be aware of what is, of course, biological null hypothesis, alternative hypothesis, statistical and alternative hypothesis. And so with the nature of your data, so for step 4, you have to consider determining the variables that are relevant to the question and determine what kind of variable each one is. So you must also know if your data is quantitative, qualitative, whether your data is nominal, ordinal, interval, or ratio. So we are going to discuss all of this one by one in a separate video. And then number 6, you also need to design an experiment that controls or randomizes the confounding variables. Number 7, based on the number of variables, the kind of variables, the expected fit to the parametric assumptions and hypothesis to be tested, choose the best statistical test to use. So as again mentioned in this step, and also mentioned in the previous step, you must know the nature of your variables, even in terms of the number. So because there are several statistical tests available, and in order for us to use whether we are going to use a parametric test or non-parametric test, so we need to check if it satisfies the conditions or assumptions of the test. Number 8, if possible, do a power analysis to determine a good sample size for the experiment. So there are also existing methods in determining for a sample size. You can also make use of the normality test later for you to determine if the condition of the test is satisfied. So for the power analysis test, we actually have the G-power test. We can also make use of the usual method that used in statistics, or some or also you might want to consider that your data must be sufficiently large enough, and it is equivalent later if in case that you want to come up with comparison. So it of course, it is dependent on your research problem. And then for number 9, you have to do the experiment. Number 10, examine the data to see if it meets the assumption of the statistical test that you choose. So as mentioned again, so like normality, homoscedasticity for the test of measurement variable. So if doesn't, choose a more appropriate test. Number 11, apply the statistical test that you choose and interpret the result. For step number 12, communicate your result effectively, usually with a graph or table. So as we can see here, we have 12 steps, which is considered by the author as the best for him to describe, for him to analyze the biological experiment. And I also want to emphasize that step number 9 is the experiment. So which means in starting or in conducting data or in conducting later research about biological data, so you do not need to start with number 9, do the experiment before coming up now or realizing now what is the questions that you want to answer. You you always must start with what is the biological question that you are asking. So that after that, it will not complicate everything as if that you do not know what will be the answer or what is the data all about. Okay, so let's have also an example presented by the same author. So this is from Verelli and Ernest in 2001. So they measured glycogen content in Drosophila melanogaster individuals. So the flies were polymorphic at the genetic locus that codes for the enzyme phosphoglucomutase or PGM at site 52 in the PGM protein sequence. Flies had an either valine or an alanine. So at site 484, they had either a valine or leucine, all four combinations of amino acids. So VV, VL, AV, AL were present. So for the step 1, so what is the biological questions that determined by the author? So one biological question is, do amino acid polymorphism at the PGM locus have an effect on glycogen content? So that's for the step 1. And then for step 2, they constructed a biological null hypothesis. So the biological null hypothesis is different amino acid sequences do not affect the biochemical properties of PGM. So glycogen content is not affected by the PGM sequence. So the biological alternative hypothesis is different amino acid sequence do not affect the biological properties of PGM. So glycogen content is affected by the PGM sequence. Third, so aside from biological, we also mentioned that there is a need for statistical. So for the statistical null hypothesis, flies with different sequences of PGM enzyme have the same average glycogen content. While for the alternative, flies with different sequence of PGM have different average glycogen content. So if you notice, when we are using a null hypothesis or alternative hypothesis, for null hypothesis, we emphasize that there is no effect, while for alternative, of course, it is the opposite of the null hypothesis. And also, if you observe that for biological null hypothesis or biological hypothesis in general, compared with the statistical hypothesis. For statistical hypothesis, we have the quantity or the numbers. So just like here, we made mention of same average glycogen content. So we are going to measure. So there is a need rather to measure for the average glycogen content. So it needs a statistical data or quantitative data. While for biological, it is a very general. It is just stating that different amino acid sequences do not affect biological or rather biochemical properties of the PGM. So how to measure that? So it is already stated in its statistical null hypothesis. Okay, let's move to the step 4. So the determined relevant variables by Verelli and Ernst are glycogen content and the PGM sequence. Step number 5, glycogen content is a measurement variable. So if we will just recall what is step number 5, it states that we have to determine what kind of variable each one is. So here, for the glycogen content, so it is a measurement variable, something that you record as a number that could have many values. So the sequence of a PGM that a fly has is BB, VL, AV, or AL. It is not numerical, it is a nominal variable or categorical variable in general. So something with a small number of possible values, 4 in the case of usually record as a word. Okay, let's move to the next one. Step number 6. So you have to design an experiment that controls or randomize the confounding variable. So For step 6, other variables that might be important. So they identified the age. So, and were in a vial, the flies pupated were either controlled, flies of all the same ages were used, or randomized. Flies were taken randomly from the vials without regard to where they pupated. So it also would have been possible to observe the confounding variables. So for example, they could have used flies of different ages and then use a statistical technique that adjusted for the age. So they consider age and were in a vial the fly pupated. So they were either controlled as mentioned or randomized. Number 7, so because the goal is to compare the means of one measure variable among groups classified by one nominal variable, and there are more to categories, the appropriate statistical test is one-way ANOVA. So once you know what variable you are realizing or analyzing rather, and what type they are, the number of possible statistical tests is usually limited to 1 or 2. So we have, again, a separate discussion on how the authors identified that the appropriate test is one-way ANOVA. And it is also based on its objective and also on the type of variables that we are going through later as we go on with this subject. And then step number 8, so a power analysis would have required an estimate of a standard deviation of the glycogen content, which probably could have been found in the published literature, and a number 4 effect size, so the variation in glycogen content among genotypes that the experimenters wanted to detect. So for power analysis, if you're going to use G-power, you can support it with the use of other researches conducted similar to the study being conducted. So you can actually make use of the effect size of other study by considering now the standard deviation, and also for the population they were, or sample they were able to use. And you can also pattern that for you to come up now with this study. So in this experiment, any difference in glycogen content among genotypes would be interesting. So the experimenters just use as many flies as was practical in the time available. Step 9, so you have to do the experiment. So the experiment was done. Glycogen content was measured in flies with different PGM sequences. Step number 10, examine the data to see if it needs the assumptions of a statistical test you chose. So primarily, so you have to check for normality, homoscedasticity, and others. So for number 10, the ANOVA assumes that the measurement variable, glycogen content, is normal. So it means that it is normally distributed. The shape of the distribution is a bell shape. And the homoscedasticity, the variance in the glycogen content of the different PGM sequences are equal. And inspecting histograms of the data shows that the data fits this assumption. So as mentioned here, you can also make use of histogram for you to determine whether the shape of your data is normal or not. So if the data had not met the assumptions of ANOVA, then there is a counterpart rather in a non-parametric, which is non-parametric. And in the case of one-way ANOVA, so we have the Kruskal-Wallis test. So step number 11, the one-way ANOVA was done using a spreadsheet, web page, or computer program. And the result of ANOVA is a p-value less than 0.05. So the interpretation is that flies with some PGM sequences have different average glycogen content than flies with other sequence of the PGM. So we have here step 12, communicate the result. So the result could be summarized in a table, but a more effective way to communicate is with the graph. So we have here the graph and the interpretation. So glycogen content in Drosophila melanogaster. Each bar represents the mean glycogen content of 12 flies with the indicated PGM haplotype. So the narrow bars represent 95% of confidence interval. So as you can see here, you also need to test or to check some assumptions first before deciding what statistical test you are going to use. But the first step is always to start with the biological question you are asking. So you have first to come up with what is your research questions or your biological questions before coming up with all of the whole process and reaching number 12, which is to communicate the result. Because of course, you need to answer your research problem. So your main objective is for you to come up with an answer to your step number 1. So in step number 12, you were able to answer your research problem or biological question in step number 1. So for the reference materials, so we have, of course, the studies used as an example, the study of Verily and Inst in 2001. So this is the study cited by McDonald's JH and the author of the Handbook of Biological Statistics, third edition. So it is published in 2014. So thank you for watching.